Notes, this is where we ended on yesterday, whatever day that was.
So far away. It seems like yeah.
So we were talking about the different layers of the eyeball, and we had the outer fibrous tunic formed of the cornea and the sclera, 114, and then we had the vascular tunic, which had the choroid layer, the ciliary processes, muscle, iris, and then we had finished up with the nervous tunic, which, if you recall, grew out of the diencephalon and created this cup-shaped structure, which had the outer pigmented area and the inner layer of nerve cells. Okay, and we kind of finished up with this picture. So we're going to look today at two different things. We're going to look at the physics of focusing, and then the physiology of the photoreceptors and the neurons and how they create the electrical current or inhibit the electrical current for sending action potentials back to the occipital lobe. So those are the two major focuses that we're covering. So if we take this area right here, that's kind of what's being illustrated over there on the board, and the pigmented cells, also known as the retinal pigmented epithelium, contain melanin, and they have two major functions. One is the absorption of light, so that it doesn't bounce around inside the eye and create additional images that would blur the focus. And the other is the recycling of visual pigments. So they'll sit there and endocytosis use opsin pigments that have been bleached, recycle them, and return them to the cell, to the photoreceptors. So to do so, they are going to have lysosomes, all right, because these are proteins that are being recycled and broken down. And so these pigmented cells are going to be high in lysosomes. So retinal pigmented epithelium, or RPE. A direct and close association between them is shown with the rods and the cones. Right, so the cones have a kind of columnar shape that is interacting with the pigment, and then a cell body and an outer area which acts, this is where neurotransmitters are released from. And cones are very similar, except that they have a cone shape. So these are photoreceptors. They're going to be generating graded potentials rather than action potentials. And as we saw in the eyeball, when we're talking about the structure of the eyeball, the fovea centralis, which is where we do our best focusing, all right? When I look at the skull of the skeleton over there, the images, light waves are being focused at the fovea centralis, and I'm seeing other objects, they're just not in focus, all right? So when I look at the skull, I'm seeing the board and I'm seeing colors on the board, but I can't read those letters until I turn and actually look at them. So right where I'm focusing on my eye, on the fovea centralis, is all cones. And as we move to the periphery, it becomes more and more rods. Until right close to the ora serrata, it's very few cones at all. Okay, way back here. Rods and cones differ in the energy of the light wave to which they are sensitive. Rods can be affected by just one photon, which is a measurement of the energy in a light wave. That's not enough energy to change anything in the cone, which is why we don't see color at night, all right, unless we turn a light on, because there's not enough energy in the light from the stars or the sun or a faint light bulb to activate the cone's pigmented visual pigments. In contrast, with a bright sunlight, there is so much energy in the light available that the rods are completely bleached out and don't work at all. And then we have in between, where some rods and cones will both be active. So when we look at this pathway, what we are visualizing is, in this pathway, these would be the photoreceptors. And we're going to come back at the very end of lecture today and follow this pathway back to the occipital lobe. Again, a repetition of what we've seen already. The photoreceptors are going to interact with the equivalent of our first order neuron, which is a bipolar neuron. And it may not be one photoreceptor for one bipolar neuron, it might be 2 or 3. So there's a variation in the ratio. So this is the equivalent of our first order neuron in a sensory pathway. And then the next cell in sequence is this cell right here. So here would be the bipolar neurons. The next cell in sequence is called a ganglion cell. And it has very long axons. And these pass out of the eyeball. And it is the axons of multiple ganglion cells that form the optic nerve. So this would be the equivalent of the second order neuron. So where the optic nerve is leaving the eyeball, you can see down here, there are no photoreceptors. And so we have a contrast. We have the fovea centralis, which has the highest focus, the best focus, best resolution, and the optic nerve, which has no photoreceptors. And so this is also known as the optic disc. The entire structure is the optic nerve. Where it leaves the eyeball is the optic disc, also known as the blind spot, because of the lack of photoreceptors as they leave the eyeball. So if light were to land on this area, there are no photoreceptors capable of responding to it. And the energy of the photon is not going to cause any action potentials to be created in these axons. So light has to pass through the axons of the ganglion cells and through or between the bipolar cells to finally reach the photoreceptors. In addition, there are other cells, horizontal cells and amacrine cells, that modulate how the bipolar and ganglion cells respond. Physiology of vision is really complex in the sense of when do we turn cells on and when do we turn cells off. Cells side by side, one may be turned off by light, one may be turned on by light, by the same light. Okay? And that affects the image and the sharpness of the edges and whether we detect motion or not. So this is just looking at the layers. So out here would be the vascular layer. Here's the pigmented layer. And this would be the outer segments that have the visual neurons and their cell bodies. And then finally we have the ganglia, the axons rather, of the ganglion cells. So light has to pass through all of these to get to the photoreceptors. Here would be the axons of the ganglion cells leaving, and as you can see, there are no photoreceptors there. So when light hits that area, there's no response. Now, at the bottom of your lecture notes on page 114, you will see an X and an O. All right, up here it's a plus and a dot. And in lab, you will be asked to measure the distance from your eye of the blind spot. The X and the O in lab are a lot larger, so that's going to change the distance. So what you want to do is, I can only close my left eye voluntarily. I have to use my finger to close my right eye. So I do this with closing my left eye. So if you're closing your left eye, you want to look at the X with your right eye. If you're closing your right eye, you want to look at the O. Okay? So I'm closing my left eye. I'm looking at the X, so the O is lateral. What does that mean about where it's going to be focused? So if this is my right eye, and here's the X and the O, and I'm focusing on the X. The X is going to be focused there on the fovea centralis. The O is going to be focused medial to that, all right? Because our images are reversed left to right, right to left on the retina, on the retinal field. So the lateral image is going to be focused on the more medial part of my eyeball, meaning medial to the fovea centralis, which is closer to the optic nerve. So as I move that, I don't know how far I'd be on the screen if I did it on the screen, but anyway, if I move this closer, so now you have to, the key to this, because a lot of people will say, I always see the O. The key is you have to look only at the X and allow the O to be in your peripheral vision. As soon as you look at the O, you're going to see it. So the hard part is to focus only on the X and allow the O to land on your peripheral photoreceptors. You'll see it, but it won't be in focus. Okay? So looking at the X, the O has now disappeared. Now I see it again. Now it's disappeared. Now I see it again. All right? So when you do that, what's happening is because the O is being, light waves are going medial, when that image lands on the optic disc, it disappears from the piece of paper because there's no ability for the retinal cells to process that because there's no retinal cells there. Okay? So if we turn this light off, this is where the optic nerve is leaving. You'll notice it's medial. So we're looking in the individual's right eye. The nose would be here. And then directly in the center, would be the view through an ophthalmoscope, is the central fovea, and then the tissue around that is called the macula lutea, or yellow spot. Okay? This is bright just because there's a light from the ophthalmoscope shining on it. There's no light coming through the optic nerve out at you. Is why it's showing bright like that. All right, so the next thing we want to do is talk about the mechanics, the physics of focusing. And so let us look at the structures and the sequence through which light passes through the eyeball. So may I erase the board? You guys okay with that? So our light source is going to be over on this end of the board. And remember, the optic nerve passes medial. So if I were to draw a line through here, this would be approximately where my fovea centralis would be. It's not quite in the midline, so it's... So I don't want to draw every single aspect of every layer, but just enough to give you an idea of the region we're looking at. So this would be the cornea and the sclera. And where the two stop, remember, is known as, the two meet rather, is known as the limbus. The vascular layer contributes to the ciliary muscle and the iris. And the lens is attached to the ciliary muscle via suspensory ligaments. Okay. Should look a little bit familiar. Now remember the ciliary processes, which were also part of the ciliary body, secreted aqueous humor. And so the aqueous humor is going to travel from those ciliary processes into the space between the lens and the iris and through the pupil. The space that is between the lens and the cornea is known as the anterior cavity. Oops, I drew that too far. It is formed of two chambers. So we would have the posterior chamber, which is between the lens and the iris. So that's the first space the aqueous humor is secreted into, is the posterior chamber. And then after it flows through the opening of the pupil, it is in the anterior chamber. So it is the oxygen content of the aqueous humor that's providing a lot of the oxygen and nutrients to the cornea. So this is between the iris and the cornea. The anterior chamber contains aqueous humor secreted by the ciliary processes. This is constantly being secreted from blood supply in the ciliary processes, the vascular layer. And because it's constantly being secreted, it has to be constantly absorbed. So remember the CSF? It's constantly being secreted. If its absorption is blocked, you get increased intracranial pressure within the ventricles. And so if this is not absorbed, we have increased intraocular pressure. Anybody know it results in blindness? Anybody know what it's called? Pardon? No? It has to do with that. It can cause retinopathy, but you can have neuroretinopathy and other causes. So increased intraocular pressure due to a blockage in the drainage of aqueous humor causes glaucoma. So if you've had your eyes examined, they'll do a puff of air. They've got some other ways of measuring it now to measure the resistance and compliance of the eyeball. All right, so my grandfather was blind, developed by the time he was in his early 80s, was blind from glaucoma. My dad has it, but his is treated. There's no cure, but usually medication helps increase and help with the absorption. And so he puts drops in his eyes every day, and he's going to be 83 in January, and his vision is, he's reading glasses, but he always has since he was a kid, but he doesn't have blindness from glaucoma. Yes?
So glaucoma causes blindness? Or?
Yes. So as a result of the increased pressure, glaucoma is the blindness caused by the increased pressure pushes back on the neurons and causes them to die, and that results in the blindness. So the increased pressure is just like if you were to put pressure on a nerve, it stops to function. gets numb or tingly. All right, so the increased pressure of this fluid not being reabsorbed results in subsequently in damage to the retina. So where this is reabsorbed is in small spaces that lead to blood vessels, small spaces in the limbus that lead to blood vessels. And these are known as the scleral venous sinuses. And so it's not known the actual direct cause, but there is a blockage in the movement of aqueous humor across the wall of these sinuses into the veins. And so these are the scleral venous sinuses. The old name is Canal of Schlemm. All right, so now behind the lens, this space is known as the posterior cavity. It also contains a material more important in helping to maintain the shape of the eyeball. And it's kind of a grape jelly-like consistency. You'll see it when we dissect the eyeballs tomorrow.